Hi everyone, I am Mauricio, the CEO of Biotech. A few weeks ago, we have an important visit in our facilities, a renowned Koen Venema, an expert in microbiome research in our facilities in Colombia. And we had a valuable discussion about the stochastic process, microbiome, mist culture, and how to produce a synthetic microbiome. So I would like to invite you to listen in to our podcast and follow us in our social media.
So they say it's like maybe it's an stochastic process. Like
Yes.
Just they change something happened
Yes.
inside and then. But
No, I mean, I can imagine that because these microbes they keep mutating, right? At a very low rate, but they keep mutating. One microorganism that you grow up to 1 million, and there's a couple of mutations in there. If that happens in a gene that is important for a certain process, and the gene is no longer functioning, then suddenly your process is different.
And how you work with that in anaerobic reactors or system when this thing happens? You need to fix it or what?
there is a very a concept that I like a lot. That is the functional redundancy.
The functional redundancy, yes.
Sometimes these changes can happen in the structure, for example, of the community, but especially in anaerobic digesters, it's a more simple process maybe. So it's always the same amount of methane that is produced, cleaning, like the
The exopolysaccharides and so on.
Yeah.
All of the metabolites that are needed
it
for
keeps
that.
working the same even when the community the structure changes, sometimes by a little. If the conditions are the same, they stay working the same. Sometimes these reactors they destabilize out of nowhere, and then you have to check what happened.
Yeah.
Okay, well, what can we do? But in the function is very very similar.
Yeah.
Even in conditions that are quite different, like different substrates, and they work. The main result of the process is the same.
So I showed one example for the Shima model, right, where you had the Bacteroides that was increased or Prevotella, but down the road, short chain fatty acids were equal.
Yeah.
Two different communities,
To different communities.
same outcome.
Yeah.
I have a biochemical training, right? So I was taught about enthalpy and entropy. Those communities try to optimize the use of the substrates, and that must mean that there is one outcome, right? Because that's the optimal one in terms of energy extraction, utilization, generation of biomass, and that's why I think there indeed is a tremendous functional redundancy. Now, having said that, last week I was in a meeting in Malta, and Alan Walker from Aberdeen was giving a talk on myths of the gut microbiota. And those plots that you see of functional redundancy are of the core enzymes that are present within each sample. So they forget about all the other metabolic activities that are not common between those samples. So he says, no, that is not really a functional redundancy. But for carbohydrates, I think there is, for sure. Because we've seen that all the time. I mean, we've done samples with 10 different stool samples from 10 different individuals. Outcome exactly the same. So there, I think there's a lot of functional redundancy. But if you, for instance, take polyphenols, there's a microbiota that can handle that, and there's a microbiota that cannot. So then there is no functional redundancy.
Okay.
Now we try to circumvent that by pooling microbiota from different individuals, right? So to pool or not to pool, that's the title of the that's the question, but that's also the title of the manuscript, the paper. Because in that way we hope to introduce microorganisms that can do a lot of different tricks. And because we model the system, we don't necessarily need exactly what happens in vivo. We just want to understand mechanisms, what occur, and then on the basis of that, try to validate that in clinical trials or animal trials. I believe that pooling is allowed. But I also understand people that say, no, you need to check every individual sample. Now, with animal microbiota, there is much more overlap between animals, right? They are co-host and things like that. So, they share microbes. So there, the individual variation is much lower.
Okay.
Yeah.
And this topic, I have been reading about the predict the microbiome, but is now the way that predict the composition of a microbiome is based on the function distribution of the data that you have.
Yeah.
But based on this discussion and for the purpose of our work, can be also be or use the same principle to try to infer a functionality in a community more than a structure in the community. Like for example, we have certain data about enzyme production or genes that are associated with enzyme production, and then we can try to train machine learning algorithm
Yeah.
to predict if the community
Yeah.
under certain condition of diet and so on
Yeah.
can produce these metabolites or this enzyme more than the structure that are composed or even both. What do you think can be also inferred? Yeah, not with all the biological data, also with in silico data.
I think so. I mean, that was my major research line at Maastricht University, to look for fibers, to look at structure-function relationship, to look at the enzymes that were present. And the idea was to use artificial intelligence to say, okay, this is my structure of the community using 16S or something like that. And then what do I need to give this community in terms of a fiber to changes to that functionality? Or the other way around, I have this fiber, I give it to a community, what happens? Does it go there? Does it go there? To be able to predict that using AI. So in Venlo, I was going to spend 15 years on that until I retire. At Maastricht University, they want to do that in 4 years in one project. So I don't see that happening, but we're also there working on that by getting that data, by understanding not only the structure but the activity of what they do there, what they produce there, and that then includes recognition of this functional redundancy that occurs. But so for carbohydrates, we look at what is called Kzymes, carbohydrate active enzymes.
Okay.
Because these are involved in the degradation of these polymers that we feed the microbiota.
Or even infer something, because in proteins they are doing something like that. Now they have even proteins or even new materials is also a field that is full of AI, and they are working. They now know based on the data set and the infer that the algorithm can do, that there are new materials that can be in certain position and so on and so forth. They need to be proved, but at least from the AI perspective, you have a candidate.
Yes.
I think, or I dream to have at certain point this in functionality with the microbiome.
Yeah.
To have some candidates of microbiome or condition that produce that functionality we want, and that can be the first filter in silico filter before to go to make some experiments
Lab
in the
experiments. Yes. Yeah, I think that should be possible in the future. The reason I think that is because we've done something, but just with a single protein. We looked at the three-dimensional structure. Based on in silico prediction, we knew what it might be doing.
Mm-hmm.
It might be cleaving a carbohydrate. We looked at three-dimensional structure. We modeled many different carbohydrates in the active pocket to predict which carbohydrates it would degrade the best. And we came to a very surprising conclusion that it was a carbohydrate that nobody uses in food a lot, but that fit best in this particular enzyme. So now this is one enzyme, and in a microbe there are 3,000
Yeah,
different ones that work together at different levels of concentrations. Some are enzymatic, some are not. Some are transcription factors and stuff like that. So all of that needs to be modeled in a proper way to be able to predict what that microorganism is doing. That's then a single microorganism. Then you're looking at a community where again you have differences in, abundance of microorganisms of several log factors. But with the current computing power that we have, and being able to compute in the cloud and whatnot, it's a matter of time before we are really able to do that. Having said that, though, when I did my PhD, one of the first genomes of lactic acid bacteria was sequenced, Lactococcus lactose. If you look at the genes, only 30% we could predict what it might do. 70%, no idea. Which is still the case. No longer 70%. We know a little bit more, but I happened to clone a gene there, and I keep the story short. But it caused a phenotype. It caused the bacteria to change color when you give it a certain substrate. It's a membrane protein. I have no idea why it changes substrate. I have no idea about its function. Every year on my birthday, I blast it against the data. I throw it into the cloud to see if somebody figured out what it did. Still not, so now we are 30 years further and nobody knows what it does. I gave it a name. It had a very similar activity as alkaline phosphatase. So I gave it alkaline phosphatase-like, APL. It's called APL.
Okay.
So
like
APL, yeah. But that is also one of the problems that we're facing. Some of these enzymes, we have really no idea what they are doing. Now, hopefully, with AI and the 3D modeling that we can do with AlphaFold and things like that,
AlphaFold
will give us some clues. But then again, this is a really difficult one because this is inserted in a membrane. So the 3D structure has to be modeled also in a membrane environment, right? So, yeah.
And for the regulatory perspective, do you think that we can have at the end like okay, this is a misculture with this probabilistic composition, or they can move around, I don't know, 21st lactobacilli, 10 of this abundant relative abundance, for example, and that can be enough to define a community, or is
I think so, because now there are also products with more than one strain, right? And they are present in that product in a certain ratio, allegedly, according to the package. I don't think that ratio is real. That is plus or minus something. So if the regulatory authorities allow that, then why not with some more complex communities as well?
and even I was thinking that when you said that some strains just grow together with other, like the co-culture or together, that is a big problem because so if you have a regulatory and you need to define the UFCs, you need to define like the CFU. Sorry.
Yeah, no, UFC is Latin America.
Yeah. Ultimate Fighting Championship. I like MMA, so that no, but yeah, I was like, how do you define this strain or the counting of cells in this strain? If it's attached or tied to the other community around. So that's why I suppose that maybe the legislation need to go to say no, is the percentage of this based on the DNA, something like that.
And then plus or minus whatever they allow, right? Because it can't be exact. That's just not possible. I know now in products, it's whatever's on the package. I had student projects that bought capsules and tablets from the pharmacy, to look at how many viable lactobacillus was in there. Some, zero.
Okay.
Nothing. Nothing. And some of which I already knew beforehand that these were good products, more than was on the package because they overdose, right? Because at the end of shelf life, what Joanna said as well, at the end of shelf life, it has to be dead. Before the end of shelf life, it's usually then higher. Now, those are good products. If there's no viable cells in there, that's horrible. And then even CFU counting is an issue because a CFU is not one cell.
Yeah.
Certainly not for lactic acid bacteria. Lactobacilli are a string of, I don't know, let's say 5 cells, right?
That's right.
Now suppose that in that string of 5 cells, only one cell is alive, you get a CFU. But only 20% of your cells are alive in that string. So even that is not really a good accurate number. Yeah, and well, don't get me going, but in those freeze-dried products, we talk about probiotics need to be alive, right? Most of the cells in these freeze-dried products are dead.
Yeah.
They are postbiotics.
Yeah, postbiotics.
So and who's what's the word I'm looking for? Why would it be that the viable microorganisms have the activity? Maybe the postbiotics are much more active, but we can't segregate that because we can't separate that. So
yeah, we need to have everything there.
Yeah. So I was asking that to Camilo. Do you also use other tools to look at your microbes than just plating? You know, do you use flow cytometry or a qPCR? Even though that's also not optimal because also dead bacteria have DNA and you amplify your dead bacteria. So we've been using a qPCR directly on the sample and after what is called PMA treatment, and I forget what PMA stands for, but it's a chemical that integrates into dead cells. It binds to DNA, and then you cannot do a PCR. So if you do it on an untreated sample, on a treated sample, then the difference is your dead bacteria.
I hear about it. Yeah.
That's very interesting. Because I've been telling them like this is a possibility to like replace the like quality control we have.
Yeah.
That sometimes we have too many samples and we don't have enough petri dishes and people to. But qPCR we can have a lot.
Yeah.
But then it's true.
It's DNA.
Yeah. DNA.
But then
this is a possibility.
But and unfortunately there is no optimal method.
This is not the solution.
No, because the PMA treatment requires UV lights to link the chemical to the DNA.
Okay.
So if you have a complex matrix like foods, the UV light doesn't go to the bacteria, but is scattered by food particles, proteins, and things like that. So it has to be bacteria in a clear solution, which in your product is not the case. Also not if you in the workshop we suspended and stuff like that. So there's limitations to everything, and I think we need to find a solution to that to get the actual counts.
That's something I said yesterday. I know it's not completely true, but I haven't found or I haven't read a convincing, reliable method to do this like counting in absolute numbers, not relative numbers. Do you have any trying test?
I would do a qPCR on total bacteria, so with primers that amplify every microorganism. That has been done. People have used flow cytometry to get the total cell count.
Yeah.
Do you know results of this?
It's hard to say. I mean, because I don't know what the truth is. I mean, the method gives me a number, yes, but is it the true number? Nobody knows. No, but with the what was I going to say with the qPCR? Usually that's done on 16S. Now, one of the problems with 16S is that You have bacteria that have one 16S copy in the genome. You have bacteria that have 14 copies of 16S in the genome. So you amplify one bacteria 14 times and another only one. That gives you a very skew in the qPCR. So you need to correct for that again. All of those things have been done, and I think it is a good idea indeed to look at that, because when you said that, my brain went back to Joanna's talk and well, the data that was shown a couple of times on the pigs with the antibiotic treatment and your product, that was relative abundance.
Yeah.
Right, and you saw also that the antibiotic resistance genes went down. That was also
Copies per million.
yeah,
But relative.
relative, right? So it could very well be that it's not the case because this was antibiotic treatment. But if there were more cells there, then the results are even better because then the number of genes per number of cells is even lower.
Yeah.
If there is a lower number of cells there, which is the case because it's antibiotic treatment, then actually the number of genes per number of cells may be the same as in your control, but because the number of cells is lower, you see also lower number of antibiotic resistance genes, right? So quantification really is something that we need to go to.
Yeah.
So complex.
We have some professor that he works with cytometry,
Yeah.
but it seems difficult to standardize the protocol for a whole microbiome, like even for pure cells, like for pure culture. They have done it, but so we don't know if we should invest in this.
Yeah.
I think that the goal is important.
Yeah. Yeah.
We need to count this in absolute numbers, so that would be very useful, I think.
Yeah, I agree. Yeah.
And I had another question, but I forgot about it. These were like 3 technical questions I wanted to make before you leave, because I don't know when we have the opportunity.
Yeah. Well, you can always contact me.
Yes.
Feel free to do so. Yes.
Thank you very much.
Yeah.
Yes. Okay, Koen, I will be leaving soon. I don't think I will go with you guys to the Mirador. Yeah, I need to pick up something.
Point of
very
view?
far.
Okay, it's okay.
Very far in the city, like Envigado. So it's a long road for me.
Okay. Yeah.
That's a big pleasure. Thank you
Yes.
very much.
Thank you very much also.